In your lab this week, we'll be looking at a fungus called Sordaria fimicola. It is a type of ascomycete. Mycetes means mushrooms or fungus, and it is a fungus that forms spores in an ascus, so it is an ascomycete. Sordaria fimicola's life cycle begins as an ascospore. These ascospores are stored inside an ascus, and a group of asci are produced inside a perithecium, or a flask-shaped fruiting structure. When there is enough pressure built up inside an ascus, the ascospores are ejected through the ostiole, or the opening of the perithecium. Once the ascospores land on suitable habitat, like decaying plant matter or dung, the haploid ascospore germinates and forms these long hyphae, which are filaments that extend and are arranged into a network called a mycelium, which gives it a fuzzy appearance. When haploid hyphae from different individuals meet each other, the cells in the hyphae join up to form a dikaryon. These cells are unique because they contain two separate haploid nuclei, so they're not actually diploid. It is only after many mitotic divisions that some of the dikaryotic cells fuse to form diploid zygotes. Then, the zygote undergoes normal meiosis and forms 4 haploid nuclei. As you have seen in your lecture notes, they undergo an additional mitotic division to produce 8 haploid nuclei. Cells are formed around these nuclei to become ascospores. When the perithecium ruptures and the ascospores are released, the life cycle is complete. Now that you have an idea of how ascomycetes propagate in nature, let's take a look at how we cross them in the lab. We can purchase different strains of Sordaria from suppliers like the Carolina Biological Supplies Company. We have here a wild-type strain, a gray mutant, and a tan mutant. With a Bunsen burner going to reduce the chance of contamination falling onto the petri dish, we cut out a small piece of the agar from one of the cultures, for example, the wild-type strain, and place it into a quadrant of a new plate. We cut out another piece and place it in the opposite quadrant from the first one. Then we do the same, but with a different strain, like a gray mutant. And we place the 2 pieces of agar in the 2 remaining quadrants of the plate. Then we place the plate upside down in a 25 degrees Celsius incubator for 72 hours. This is the result. This cross here is between wild type and tan. This one is between wild type and gray, and this one is between gray and tan. You can clearly identify the boundaries where the 2 different strains meet, and that's where we want to collect the perithecia. We take a small spatula or a dissecting needle and scoop some of the perithecia into a drop of water or saline on a glass slide. Then we place the cover slip on top. What we want to do is to use the tip of a needle to gently press onto the cover slip to force the perithecia to burst. We want to do this under the microscope so that we can see the asci coming out, and we can tell how hard we should be pressing onto the cover slip. We want to avoid pressing too hard and causing the asci to break apart and the ascospores to be ejected, because then we would no longer be able to identify their segregation pattern. Then we carefully look at the asci containing mature ascospores and count how many M1 and M2 segregation patterns we can see in a given perithecium. We can see that some are not fully mature and those do not have coloration. This one here that's circled on the screen is an M1 pattern, while this other one is an M2 pattern. The slide we're looking at contains the cross between wild type and tan. This one is wild type and gray. And finally, this one is tan and gray. Note that since we're looking at three-dimensional structures, we often have to focus up and down in order to see the spores in all of the asci, and sometimes to fully see all of the spores in one ascus. Can you find a tetratype ascus? Pause the video now and see if you can find one. If you picked this one, congratulations! You have correctly identified a tetratype ascus. Hopefully this gives you a good idea of how Sordaria crosses are prepared in lab. See you in lab!